that's what we're going to look at now. And that story is told, is that property is held by proteins called snares for historical, it turns out they were named for how they were purified. So the snare proteins provide specificity for vesicle targeting. And this is how they work. This is a generic slide from your book right here. If this were the endoplasmic reticulum, we could look at it right here. This could be the plasma membrane. So this is the donor compartment. We see, let's pretend it's the ER. So we see right here that there are coat proteins forming right here. Left out are the SAR1 proteins that are making this coat protein fall off. This coat protein assemble here to bud off a vesicle. Trapped in that vesicle would be any receptors that have targeting signals for the coat, as well as any protein that they're carrying. And so those go into the coated vesicle. We know that these coat proteins almost immediately fall off. So all disassemble into monomers in the cytoplasm. They can then recycle back to make more coat proteins. And this naked vesicle must fuse with the appropriate membrane. And the reason why an ER-derived vesicle will fuse with the cis Golgi is because of these snare proteins. So here we see a light pink snare protein, and it's binding and fusing with this compartment A, because this light pink snare will recognize only this light blue snare. And it's the coupling of those two proteins that make the fusion happen. And so we see that the vesicle carries one kind of snare protein. That's called a V snare for vesicle snare. And they will bind to a T snare or a target vesicle. Now, even from the same compartment, you may have a different V snare that's going into a different kind of vesicle. That's shown with the dark red V snare right here. And we see that the dark red V snare will fuse only with the darker blue T snare. And so this vesicle will fuse only with compartment B, whereas the light pink one will go only with T snare. So each of the vesicles that are budding from the appropriate compartment carry their own V snare that will make them recognize the appropriate T snare, the appropriate target. So the vesicles, the COP2 vesicles that are budding from the ER carry V snares that will recognize T snares on the cis Golgi network. The COP1 coated vesicles carry V snares, and as they bud off from the cis Golgi network, those V snares will recognize only the T snares on the ER, etc. So again, it is the coupling of that V snare with the appropriate T snare that makes the appropriate targeting, and we'll see fusion happen. So it's not in the coat because the coat falls off, but these V snares and T snares actually have a small transmembrane domain. They're embedded in the membrane. So once these peripheral membrane coat proteins fall off, the V snare is exposed and it's still there. And of course, the receptors that this vesicle is scooped up from the donor compartment are also there. Does that make sense? Excellent. So let's see how it works. So here we have a vesicle right here. This is a synaptic vesicle. And the V snare that it's carrying in synaptic vesicles, so remember that synaptic vesicles fuse with the plasma membrane and then release neurotransmitters. The neuron is just a specialized cell. And these synaptic vesicles, you don't have to remember these names, carry a protein called synaptobrevin, and that is the V snare. And the reason they fuse with the appropriate plasma membrane is that the plasma membrane, the nerve cell plasma membrane, the T snare is called syntaxin. And nerves are a little bit different in that they have sort of a third T snare called SNAP25. So but it's a little bit a specialty. But what you can see in this T snare, V snare reaction really nicely here is that it crosses the membrane, but it has only a little dinky cytoplasmic domain. So it's anchored in this bilayer, though, of the V snare anchored in the vesicle, the T snare anchored here. And these have very long cytoplasmic domains, and those intertwine and get closer, and as these bind, they serve not only to recognize this, but it'll bring this vesicle closer and closer and closer until fusion happens. So this is well worked out in neurons, with a V snare and a T snare being responsible for the release of the synaptic vesicle at the nerve plasma membrane. Now in this case, the V snares and the T snares are regulated in ways so that the synaptic vesicle will fuse only at the appropriate time. This fusion is so tight that you actually need accessory. So once the fusion happens, so here we have the V snare and the T snare, bringing this vesicle closer and closer together until we have fusion happen. And it's so tight that we need an accessory protein called NSF to actually unwind these proteins. And NSF requires ATP to get these V snares and T snares separated from each other. So it needs the energy of ATP to twist these apart, and then the V snare can be recycled back to the appropriate vesicle membrane. So how do these cause fusion? This is hypothetical. It's been modeled. Of course, we can't see this kind of thing. But here we have a vesicle right here with the V snare coming together with the T snares, and these twist. They bind really tightly and twist, 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 until the vesicle gets so close together that the two outer bilayers will spontaneously form, and you get these forming together until fusion happens, and you have a complete melting, a complete melding of the vesicle bilayer with the target membrane. Okay, so it turns out that this story with the V snares and the T snares actually is the molecular principle that Botox works on. So how many people have heard of Botox? Probably just about everybody. It's pretty, you see billboards for it, you see ads on TV. And what Botox is it will paralyze muscles. And it's injected into the forehead and sometimes around eyes, so that you get, instead, if you wrinkle your forehead, so people develop wrinkles with aging, of course, and then if you have Botox, you actually become, well, some might say younger looking. I think this guy's sort of weird looking. But as much as he raises his eyebrows as a journalist who underwent Botox, there's no wrinkles there because his forehead is paralyzed. And it turns out that Botox is actually a protease. So Botox is derived from botulinum toxin, which is lethal. It grows in improperly canned foods, in the anaerobic environment of that. If you ate those improperly canned foods, you would die because your diaphragm is paralyzed and you can't breathe. But in minute doses put into the forehead, it'll paralyze these muscles much the way Botox is. Much the way botulinum toxin will paralyze your diaphragm if you eat it. So it's a protease, it turns out. And it's a very specific protease that will destroy SNAP25, one of the T snares here. So what we have with Botox is we've destroyed the T snare here. And so this vesicle that containing the neurotransmitters, cannot fuse with the nerve cell plasma membrane, and you end, because it can't, through the acetylcholine or whatever the neurotransmitter is here, can't fuse. You have all these neurotransmitters building up, and you can't contract that muscle. As hard as he tries, his forehead muscles will not contract, and because they're not contracting, he lacks all facial expression, just about. So, sort of an interesting little story of how Botox works, something that you can share with your families or your next party that you're at, when there's a moment of conversation about Botox, about these sneers and T snares. I recommend you all go and do that in your social life. Okay, so that's where we're going to end today. We're going to start the next class with an Instapoll question, and then we'll start talking about what happens in the ER and targeting signals that go throughout the investigative hour. Again, please study your notes within 24 hours.